I'm Pippa Corrie and I'm a medical oncologist in Cambridge, and I'm going to talk to you a little bit about melanoma clinical trials in the UK. So for those of you who may have any form of cynicism towards why we do research, I just want to remind you that research drives healthcare. So here we have the Duke of Cambridge receiving his COVID vaccine. And just to remind you that really in the last 18 months, there's been a massive undertaking between academia, industry, and society as a whole in investing and delivering and developing the COVID vaccinations that have really transformed how we are managing the current COVID pandemic. Now, we've had a similar revolution in the last 10 years associated with developing treatments for metastatic melanoma patients. And here in this survival curve at time 0, you can see that 100% of people are alive to begin with. But unfortunately, over time, many people will die of their disease. And in the old days with conventional chemotherapy, most people would die within 2 or 3 years of diagnosis. But what this curve shows is that there are a series of lines that are rising over time, suggesting that more patients are living with their disease. And this is a really impressive program of clinical trials with different drugs being tested systematically and being shown to make an impact and improve outcomes for our patients. So how are drugs developed? Well, it's a complex and expensive and time-consuming business. We start with a lot of compounds that are being tested in the laboratory, and only a small number will get through to being tested in people. We first test them for safety and then for efficacy, and then ultimately they're tested against standard of care. So only a small number will actually enter the clinic. But it is expensive, and the average drug development cost is around £1 billion. And the average market price is about £100,000 per annum per patient. So you can see how expensive it is to treat people with modern treatments. And this is just for cancer, but drugs are being developed across all diseases. So what is the problem with melanoma? Well, many people will have surgery, and 80% of people will be cured by that surgery, which is fabulous. But unfortunately, for about 20% of people, their cancer will return. And this can return locally in regional lymph nodes, or it can spread to places like the liver, the lungs, the liver, the brain, places that surgeons can't get to. So people like myself have been working for the last 20 years or more to try and develop better treatments for this disease. And we tend to start by testing these drugs in what we call stage 4 metastatic melanoma patients. And the goals of treatment here are palliative, and by that we mean trying to prolong survival and improve quality of life. Now, if we find treatments that work, we can try and test them earlier in the patient pathway. So here we have patients who have regional lymph node disease that the surgeons will remove, but they have high risk of recurrence. So we can use drug treatments in what we call adjuvant therapy to try and reduce that risk of recurrence. We can potentially put those drugs before surgery and call that neoadjuvant therapy. And we can test adjuvant therapy in earlier stages in stage 2 melanoma. Even more importantly, though, in early stages is actually prevention. Prevention is always better than cure. And many people will be familiar with the slip, slop, slap program of cancer screening, of sun screening to protect against sun damage. But also we want to look at screening and early detection programs to try and enhance our ability to prevent the disease. So we call this primary prevention. And for those of you interested in other terminology, we call that adjuvant treatment secondary prevention, trying to prevent metastatic disease. And ultimately, our real goal is to convert not just palliative approaches, but to actually curing people. So what are these drugs that have been tested against melanoma that have really revolutionized our management today? Well, first of all, we have immunotherapy. And this is a cartoon of a T cell, really the key cell which is involved in killing off tumor cells in our body. We call that immunotherapy. And then we have about 40% of melanomas that have something called a BRAF mutation. And this gene mutation drives a particular pathway that is driving the tumor cell growth. And this we call BRAF targeted therapy. But let's start and talk a little bit about immunotherapy, and to try and put this in very simple terms. We're really interested in driving that T cell response. Now, T cells are white cells in our body that fight infection. And activated T cells generate inflammation. Now, T cells distinguish self from non-self. And in this diagram here on the left, you can see self. So in our body, we have circulating T cells, but they don't attack our body organs because they are self. But if a parasite or a coronavirus enters our body, then the T cells recognize that as being non-self and get to work, generate inflammation, all about trying to kill off those abnormal cells. So why doesn't the T cell do the same thing with cancer cells? Well, the problem is that cancer cells are derived from self, so they're not readily detected by the T cells. And also cancer cells can evolve mechanisms to suppress and evade the immune system. So here's that T cell that we looked at before. And what you can see is the T cell is busy interacting with its surroundings. It has a series of molecules on its surface, some of which have an inhibitory function to try and stop that T cell working, and others that are actually busy trying to activate that T cell. So there's a balance going on all the time. Now, this gentleman over here, Jim Allison, won a Nobel Prize in 2018 for working out that if you take a molecule called an antibody to block this particular receptor called CTLA4, then you could actually drive that T cell into being really active. And we call this taking the brakes off the immune system. And this is the basis of what we call the immune checkpoint inhibitors. And the first of these drugs, called ipilimumab, that entered into a clinical trial of metastatic melanoma patients, was able to demonstrate that actually over time, although a number of patients did succumb to their disease, what you see in this survival curve here is this plateau, and this flattening off is really important in demonstrating that about 20% of people didn't succumb to their disease. And in fact, after the work with developing the CTLA4 antibodies, it became apparent that if you blocked this receptor, PD-1, you could get a better survival benefit. And even combining these two antibodies together, you could improve outcomes further. But it's not all plain sailing. The immune checkpoint inhibitors also have a number of complex side effects, and these can be both life-changing and life-threatening. They are inflammatory type processes like colitis, pneumonitis, hepatitis. All the itises reflect that inflammation, some of which can be really serious, like the colitis, and if the myositis affects the heart, for example. Some of these things are reversible, but some of the damage to the thyroid, to the pituitary gland can be permanent and affect things like fertility in young people. So there is a multi-organ effect of these drugs. So when it comes to how we are trying to develop these treatments more in the clinic, we're designing clinical trials to understand how much treatment is needed, how can we predict who will respond or have toxicity, and what happens if these checkpoint inhibitors don't work. So the first trial I wanted to highlight to you is the Dante trial, which is evaluating the duration of anti-PD-1 antibody treatment in patients with metastatic melanoma. And this is because increasing experience suggests that long-term treatment is not needed for all patients, and that we know that the longer we treat, the greater the risk of toxicity and the added NHS burden with the resources needed to treat. So in patients who've been progression-free after 1 year of treatment with anti-PD-1 plus or minus CTLA4 antibodies, so conventional immunotherapy, we're inviting those patients to be randomized either to continue their treatment for a further year or to stop treatment and be followed up, but have the option to be retreated if their cancer comes back. And that trial is ongoing in many centres across the UK currently. Now, you'll have heard already today about the importance of the gut flora and how this appears to influence our immunity. We in Cambridge have been working with colleagues at the Wellcome Sanger Institute for a number of years and have been collecting stool samples from metastatic melanoma patients receiving immunotherapy. And collecting that data together, it does appear that there may be a group of bacteria that are clearly associated with response to immune checkpoint inhibitors. Now, we're not the only people to be working in this space. There are a number of different groups who have published data suggesting key bacteria are linked to the immune response, but none of them are consistent. They all have proposed different bacteria. What we're interested in or impressed by with our bacterial consortium is that we can actually apply our data across the published sets of data, and we can differentiate responders and non-responders using our bacterial consortium. And this is not just across melanoma patients, but also kidney and lung cancer patients receiving immunotherapy. So we now have a program grant with Cancer Research UK supported by Microbiome, and we are recruiting patients to MITRE, otherwise known as the Microbiome Immunotherapy Toxicity and Response Evaluation Study. And we're recruiting melanoma, renal and lung cancer patients receiving conventional immunotherapy and asking patients to collect and provide us with some stool samples prior to treating and during their treatment so that we can validate that bacterial biomarker, both in terms of its ability to predict for response and toxicity, but also potentially as an adjuvant alongside immune therapy to try and improve outcomes ultimately. So what happens if the checkpoint inhibitors don't work? Well, you can see on this T cell that there are lots of other receptors around here, and all of these are potential targets that are being evaluated for their ability to improve outcomes for our patients. But I just wanted to touch on a new concept. Well, it's not actually that new concept, but it's a relatively new concept in the UK, where we're actually taking tumours out of patients, getting our surgeons to work with us and resect tumours. And those tumours are then being sent to the laboratory where we're extracting the immune T cells and growing those up in a laboratory and then returning those back, large volumes of them, back into the patient to see if we can really boost their immune response. And we call this cell-based therapy or TIL therapy. And the first of the trials that we have been involved in the UK with a company called Iovance recently published the data. And this was taking a group of heavily pretreated patients. And 1/3 of those patients had a really good response that was durable over months and years. So that's quite exciting. And there are a number of other similar studies that are either recruiting, like the Achilles Theta study, and another study in setup called Instill Bio's Delta 1 trial. So this is a new way of working, but it does require that patients have accessible tumor that can be removed for this kind of process. So back to those patients who have a BRAF mutation. So the BRAF gene is basically DNA, and that DNA in the tumour is actually secreted into the circulation, and we call that circulating tumour DNA that can actually be measured. And we're interested to know whether that ctDNA can be useful in guiding treatment. And the team in Manchester, the Christie Hospital, have expertise in this area. And particularly in BRAF mutant metastatic melanoma patients, there is a big issue here. They actually have the option to receive immunotherapy or BRAF targeted therapy. And we don't really know what is the right schedule, which we should use first. So the question in this particular trial, known as CACTUS, Circulating Tumor DNA Guided Therapy Switch, is about using that ctDNA being measured in blood samples to try and guide which is the right scheduling for our patients. I've talked a lot about metastatic melanoma, but as I mentioned earlier, if we have effective treatments there, we use them in the adjuvant setting to try and improve outcomes of those patients who have had their earlier stage disease resected, and we're trying to avoid them developing metastatic disease. Many UK patients have been involved and have taken part in really groundbreaking adjuvant trials that have been international studies. The COMBI-AD trial evaluating dabrafenib and trametinib in BRAF mutant melanoma, and the KEYNOTE-054 trial is one of several immunotherapy trials, and have really demonstrated in these survival curves. You can see the top curve in each case is the treatment arm, the bottom curve in each case is the standard no treatment arm, and you can see there's big space between the two curves, i.e. the treatment is really making a difference and improving outcomes and literally halving risk of recurrence. So very recently, in the last month, again, an international trial which involved UK patients was published, and this was in stage 2 melanoma, so an earlier stage of disease. And again, the immunotherapy drug here, pembrolizumab, was shown to reduce risk of recurrence. But what you see here in this survival curve is you can see the brown curve is the standard no treatment arm, and the green curve is the treatment arm. And you can see that the treatment is on top. Those patients do better, but the gap between the curves is much narrower. So there's a very statistical p-value. So from a statistical perspective, it's very positive. But from a patient perspective, we need to think about what this actually means. So first of all, you can see that a high proportion of patients are going to live and never have recurrence without any treatment. So if we give treatment to everybody, we're going to be over-treating a lot of people and exposing them to potential unpleasant side effects of treatment. So the question is, should we be waiting and keep that treatment back until the time of recurrence? So an important trial, again, that the Christie team are leading on focuses again on the use of circulating tumor DNA in those patients with resected stage 2B and C melanoma. And in these patients, at the time when the ctDNA is detected, patients will be randomized either to receive immunotherapy or actually to continue to be monitored until that time, if it were to come, that is, of clinical and radiological relapse. And that will be a really interesting study to understand how we can really focus the right treatment on the right patients. And another trial that is in setup at the moment is actually trying to use that concept of bringing that adjuvant therapy before surgery. And we can do this in BRAF mutant melanoma patients because we know that BRAF targeted therapy is so effective and has a very high response rate in the early setting. So in this trial, patients will be randomized to receive 8 weeks of their adjuvant therapy before their surgery. Here, encorafenib and binimetinib, which is a BRAF MEK inhibitor combination. And the primary goal of this trial is to look at complete pathological response at surgery. But we'll also go on and have a look at how this correlates with relapse-free survival and compare it to a group of patients who have standard adjuvant therapy. And that study is again up in setup across a number of centres in the UK. But before I finish, I just wanted to touch on a rarer group of patients with a rare form of melanoma called ocular melanoma. And this drug called tebentafusp is an immunotherapy drug developed by a biotech company in Oxford. And a number of us were involved in the early stages and throughout the development of this drug. So many UK patients have been involved in its development. And in ocular melanoma, standard immunotherapies have much, much lower response rates than in cutaneous melanoma. So a trial was undertaken to compare treatment with tebentafusp against investigators' choice, which could be immunotherapy or chemotherapy. And you can again see a very big difference in overall survival of the two groups. And while the company is going for licensing for this drug, they are making it available in an expanded access program in selected sites in the UK. So that's very exciting and really great to demonstrate how UK clinicians and scientists and patients can all be involved in the evolution of new treatments. And just to finish here, just to remind you that you yourselves can go and find out more about clinical trials. There are a couple of websites to draw your attention to. The National Cancer Research Institute has a website open to the public with portfolio maps, one of which is dedicated to melanoma trials. And the National Institute for Health Research has a patient-orientated web page called Be Part of Research, where you can plug in a disease and your postcode and identify trials that are available near you. And importantly, Melanoma Focus is trying to take the work away from you and develop our own trial finder, which is it's work in progress at the moment, but hopefully will be available in the not too distant future. Thank you.